In DNA replication, where the process of transcription is DNA to make RNA, translation is the second major portion of it, which is RNA to synthesize proteins. Now in translation, there are a few words that we also need to be very familiar with. Our genetic code is our instructions needed to convert that DNA into the actual proteins. If you think back to when we covered macromolecules, remember that those polypeptide chains of amino acids eventually start to link together to make the actual protein. The combination of the various amino acid chains make the protein. When we talk about these nitrogenous bases, we usually refer to them as being in groups of 3. When they are 3 consecutive nucleotides, we call that a codon. And a codon is what's going to be used to make a specific single amino acid in mRNA. The anticodon is going to be the opposite side, the one that tRNA comes and links to on the mRNA. And translation obviously being the whole process of protein synthesis, where we use mRNA attach tRNA to make the process of the chain, which eventually is going to be brought together to make proteins. That is our translation portion of DNA replication. Amino acids are the actual structure, the building blocks, the monomers that make all the proteins. The ribosome is the organelle in the cell where this takes place. And I mentioned to you how tRNA is going to start linking amino acids together in a long chain. We call that a polypeptide chain. So it's a group of all these amino acids joined together. And I said, when you join several chains together, you make a protein. Now, if you remember this wheel here from back in macromolecules, this wheel is kind of like the code that we use, the chart, if you will, to make the amino acids. And it shows the 64 different ways to make our 20 different amino acids and our stops as well. Remember that the wheel is read from the inside out, from 5' to 3'. And there are, for example, in serine, this particular amino acid, there are 4 different ways to make, there are 4 different codons, I should say, that make that amino acid. Uracil, cytosine, and any one of the other 4 repeated again. So UCA makes serine, UCG or UCC or UCU. Some of them have 4 different ways, 4 different codons to make this amino acid. Some of them only have 2 different codons to make that particular amino acid. Just keeping in mind that this wheel, which is universal, it just basically shows all the different codons that code for a particular amino acid. You don't have to memorize it in terms of the EOC, but you do need to understand how to read it. And so we read it from the inside out to give you the codons that would make a particular amino acid. Remember that the properties of a protein are determined by the order in which these amino acids are going to be joined. If you look back to the previous slide that I showed, you'll see how different amino acids were joined in a particular order on that polypeptide chain. That would make a particular protein. If you change the order, it would make a completely different protein. Here, the chart gives you the actual names of the amino acids, which is why I included it there. Again, for the purposes of the EOC, you don't need to know it, but if it's given as a choice on the test, they would clearly give you, like for example, this one, this one, this one, and this one. Because of the fact that they start with different phonemes, different letters, you would be able to differentiate between the four. Okay? The language of mRNA is what we refer to as that genetic code. Remember that in RNA, thymine is replaced with uracil. So you don't see a T anywhere. This is coding for mRNA. So all thymine was replaced with uracil. The codon consists of the 3 nucleotides that are going to specifically be in a particular order to make that amino acid. They're going to be linked together to make that polypeptide chain. There are 64 possible 3-base codons that we come up with to make our 20 amino acids. So now look at this following RNA sequence. Let me make this disappear again. When you look at this, we're going to break this up into groups of 3. So this sequence here would actually appear like this. Now, based on this sequence, what would be the codons for the following amino acids? Or, sorry, let me rephrase that. These codons would code for what amino acids? Which 3 amino acids would be represented here with these 3 codons? You use the wheel to answer the question. When you take a moment and look, obviously since we know we read from the inside out, we see UCG, you come here to U, UCG makes what? Serine. So, CAC, you come here to CAC, you see CRA, histine, and so forth. And this is how you read. You're gonna get these 3 amino acids based on these 3 codons. Okay? Again, just another exercise for you to make sure that you understand how to read the wheel from the inside 5' to the outside 3'. All right. Again, in a nutshell, translation is that decoding of mRNA to make the polypeptide chains of amino acids. During translation, the continuation of these linking of amino acids forms the chain, which eventually linked together is protein synthesis. Protein synthesis is another way of saying translation to a certain degree. We take mRNA to eventually produce proteins. Remember, transcription is DNA to RNA. Translation is RNA to protein. So translation could be thought of as protein synthesis. The cell uses all 3 types of RNA during translation. mRNA is used because that's the initial copy that leaves the nucleus and goes over near the ribosomes. tRNA is going to attach codon to anticodon to start forming the polypeptide chain, and ribosomal RNA is the RNA at the site of the ribosome that's going to be used to make the protein. So all 3 forms of RNA, mRNA, tRNA, and rRNA are used during translation. DRNA serves as the template to RNA polymerase. RNA polymerase is the enzyme that reads the whole process and starts to make the copy of the RNA, of the mRNA, that's going to eventually leave the nucleus here, exit out that nuclear membrane, and come out this way. tRNA is going to come and attach the anticodons to it and then go over to the ribosome. Remember that when they attach, they break off in sections of 3. The codon and the anticodon will break off and basically link to that chain. It will break off the mRNA and link through tRNA to make that chain longer and longer, until eventually it reaches the point where it has reached its capacity, if you will, necessary to code for that protein. So RNA polymerase, which is probably considered to be the most important of all the 4 enzymes, is going to create the mRNA molecule. That mRNA enters the cytoplasm and binds to the ribosome. tRNA brings one amino acid at a time, links to it, and the amino acids are going to be added. They basically start popping up along the top of tRNA, and they basically do that, like I said, until that stop codon is reached. Once that happens, that's it. The polypeptide chain is as long as it needs to be to code for a particular protein. What is this? Okay, hopefully this is not going to cause a problem. Now, during this whole process of transcription and translation, I had mentioned to you before that sometimes errors can occur. And we're going to go over that now a little bit in discussing mutations. In discussing mutations, there are a few words that we should be pretty familiar with. The suffix gen means something that produces. So a mutagen, for example, is an agent that is going to induce or increase, which we could say produce, a mutation. A mutagen is that agent that is going to produce a mutation. Chemicals, UV rays from the sun, radioactive material, those are, like, for example, the 3 biggest types of mutagens that we have. Carcinogens are things that increase, and there's that suffix gen at the end again. Carcinogens are those materials that are basically going to increase the risk of cancer, like nicotine, for example, is a huge carcinogen. I included this picture here so that whenever you get a chance, you can see the effects of UV rays and how they have serious detrimental effects on your skin. So whenever you get a chance, since you guys are at an age where you like to go to the beach a lot and you like to get a lot of sun and you don't put on suntan lotion and protect yourself with a good SPF and so forth, take a second when you get a chance and read how this kind of stuff can catch up to you later in life and increase your chances of skin cancer. All right, so let's talk about mutations a little bit. And like I said, we're going to go through some major mutations that typically occur. When you read the following 2 sentences, what do we have here? There's basically one little difference. This T for this R. But the fact that we only changed one letter in an entire sentence did what? It changed the entire meaning of the entire sentence. And in fact, it made it to a point where just changing one letter in an entire sentence made the sentence not make sense. Well, in terms of genetics, the same thing happens. During mitosis, you have an error like this, and during DNA replication, when you have an error like this occur, the end product could end up being a product that, for the most part, doesn't make sense. Changing this T to an R, the dog bit the cat, as opposed to the dog bit the car, you know that that doesn't really make sense, whereas to the first one does. This is a clear example of a mutation. Okay? And again, once this error occurs during DNA replication, when that cell continues to copy, every daughter cell derived from that parent cell is going to include that copy. So that mutation is just going to increase the chances that that mutation will appear in the offspring. The words in the sentence here are similar to how an mRNA molecule is going to be lined up. Notice how I included what? 3-letter words. So how's the similarity there? They're all like codons. And in only one codon was there a change. And it was only a change in one of the nitrogenous bases, in one of the nucleotides. All right, but that right there makes the entire polypeptide chain basically change in its course of action. So what happened when a single letter was changed? The whole thing changed and didn't make sense. If you change any other letter, will the sentence continue to make sense? Can I repair this error by changing something else somewhere else? No, the error is only going to be magnified even more. When we did human reproduction earlier, I told you that 25%, 1 out of every 4 women, will miscarry from births that are started. This is typically, and again, this is just a theory, but this is typically the reason why we believe to have that happen. Okay, it's errors like this. You cannot, fix a wrong with creating another wrong. What would happen to the sentence if you delete the letter G? So if we delete the letter G, obviously we lose the codon here. What would happen? Now we would have something, instead of a dog, it would be the do bit. Obviously we see that grammatically speaking, it just gets even worse. So that's omission, for example. That's another one of our errors in mutation that makes matters even worse. Not to say that some errors are worse than others, but some errors can have greater impact on the offspring than others. So, like I said, cells are going to make mistakes. You can either, for example, you can insert, you can skip, you can do things like that are going to create mutations in the genetic materials. These are known as gene mutations. Okay? Mutations are going to produce changes in the entire chromosome, and so we refer to that as chromosomal mutations. When it gets to this point that it will show, obviously that's a clear indication of a mutation in the chromosome. Mutations in eggs or sperm during the process of mitosis, in other words, in process of meiosis, are going to affect future generations, producing the offspring with that mutation to appear. it could result in the embryo not surviving, that 25% that I spoke to you about, and it may provide an organism with an advantage. There's where we talk about natural selection. The genetic diversity is where sometimes I spoke to you and I told you that some mutations are good and some mutations are bad. In the sense where a mutation is a good mutation, that is basically evolution. That is leading toward natural selection, where the organism is developing genetic variability to enhance the mutation, because that mutation enhances its chance of survival based on changes in the environment. Mutations in body cells affect only the individual, like, for example, in the term of cancer. All right, so some of our mutations that are most common. You have, for example, a substitution. In the case of a substitution, you're supposed to have GA, but then this G right here, this A right here gets substituted by a G. Therefore, this T is going to be substituted by a C. This changes the sequence. So substitution is going to do what? It's going to create a mutation that's going to eventually lead to chromosomal mutation that's going to probably appear in the offspring. You have deletion, where it completely deletes. You have GA, it completely deletes this GA right here, and it just brings it down to 2. All right, that's going to show as well. You have insertion, where you go from GA to GA, and then it inserts an additional GA in there, in between the original ones. That would create a mutation. And you have inversion, where you flip. Not only do you flip the GA, but you flip it down to the bottom, and you flip from GA to AG. Okay? That would be inversion. These are the 4 biggest types of mutations that you'll see. So make sure you get a chance to learn what substitution looks like, what deletion looks like, what insertion looks like, and what inversion looks like. Okay? The significance of mutations. Remember, I told you that there are good mutations and bad mutations. In the case of most mutations, many of you have mutations inside of you. But sometimes these mutations can be so insignificant and so small that we consider them to be neutral. They have little or no effect on the expression of how it would show in your phenotype, physically on you. Or to the point where it really wouldn't impede any of your functions, nor the function for you to be able to process, or your body to be able to process protein synthesis, which obviously is extremely important and linked to the process of mitosis. Now, mutations that cause dramatic changes in protein structure are going to be, eventually detrimental to your health, and can basically include or lead to a disruptive normal lifestyle in terms of biological activities. Down syndrome, for example, is probably one of our most popular one. Either an extra chromosome, a missing chromosome, and all this can occur when, remember, these certain proteins make an error. And when that error occurs in lining up chromosomes, once that copy occurs, remember, it's like me going to a copy machine, taking a paper. I've made the copy of this paper, then all of a sudden I pause the copy machine, I take the paper out, and I just draw a black line on the paper. That's a small little mistake. And then I put the copy back into the copy machine and continue making copies. Every single copy does what? It just continues with that black line on it. If that's the cell that ends up, coming out during fertilization and so forth, that copy is going to show, obviously, in the offspring. And then mutations can also create genetic variability. The relation between the horse and the giraffe is the clear indication of a genetic variability that needed to occur for the survival of that organism. So there's an indication where mutation is good. Even in us, to go from the jaw structure of the nomad, the larger protruded jaw, once we stopped our eating habits the way they did, the addition of the utensils and so forth and all that, the lack of need to only eat meat and so forth, the diversification of our diet and so forth, little by little creates that jaw to do what? To shrink down in size. It didn't need to be as large any longer. So therefore, that is a mutation that for us, for our purposes, ends up being helpful. This is one of the things that on the Galapagos Islands, Darwin was completely fascinated with in terms of all the different species of finch that he found in the series of these islands, and he saw the genetic variability in the size proportions of body size and size of beak as well. Those are clear indications of genetic variability caused from beneficial mutations. Finally, let me talk to you a little bit about cancer. The chemicals in cigarettes are known as carcinogens. So take a moment to think what could lead to the development of lung cancer in a smoker. These are the pictures. This is the picture of lungs in a smoker. This is the picture of lungs in an individual that is not a smoker. And also important to know that today we've learned, and we've known this for a while now, that you don't necessarily have to be a smoker in order to develop this. You can develop things like emphysema and so forth from secondhand smoke or from dusts and certain chemicals and things like that you're exposed to based on where you work, even though you yourself do not smoke. It is clearly evident that these lungs are completely filled with tar. And if the alveoli, which are those small little sacs that open and close and fill with air to expand and contract the lung, if they get clogged with tar, then what happens? The lung itself cannot expand and contract no matter how hard that diaphragm pushes. And so therefore breathing becomes an issue. This tar, I've seen videos of individuals actually spit black in their saliva when they suffer from severe cases of lung cancer, that they just simply cough and they will spit up phlegm, and that phlegm will actually be dark colored. There's a current big issue right now, in fact, in Major League Baseball, with the addiction of what? Of chewing tobacco. chewing tobacco also maintains carcinogens in it, is known to have carcinogens. Keep in mind that the addition of carcinogens into your body is always going to increase your risks of developing cancer. Now, not really on the fence exactly where you consider UV rays as a carcinogen or not, but we also consider UV rays to be a risk factor, a huge risk factor in the risk of developing skin cancer. So there are a lot of human issues involving here regarding this. They even changed the labels. Big Tobacco, if you had to change the labels, laws were put into effect as to where they can sell tobacco. Certain stores had to be a certain distance away from schools for them to legally be able to sell tobaccos. And ads had to be a certain distance away from elementary schools and middle schools and even high schools, probably, as to where Big Tobacco was able to place an ad regarding, the sale of their product. You couldn't have a billboard. Well, you could a long time ago, but not anymore. You couldn't have a billboard right up beside, half a block away from a school promoting the sale of cigarettes. In fact, Big Tobacco, quite a few years ago, even reduced the amount of money that they spent in advertisement because of the amount of money that they've spent in legal fees and payouts in lawsuits and so forth. So whenever you get a chance, think about the effects that cancer can have on you, and Google that, if you will, and read an article or two regarding the effects of cancer on human, or links of cancer and mutations in genes. This is the conclusion of the third video of DNA. So at this point, make sure that you review all 3 videos of DNA for the class.